So without further ado, I would like to invite Dr. Lakshmi Santhanam to start her presentation, and I hope you guys enjoy.
Thank you, Sarah, for that very kind introduction, and thanks everybody for tuning in. I'm guessing you're all from various time zones, so I really appreciate you taking the time to join us today. So I am going to talk about vascular aging in the context of using preclinical models to study vascular aging, which is where our new knock-in mouse model comes in. So what I thought I would do is just go over the mechanical perspective of vascular structure and function, because the audience, I'm guessing, is a mixed audience, some of whom may be quite familiar with the terminology as well as the methodologies used. But for others, it may be beneficial to have a brief background on what vascular stiffening means and what the biophysical, biochemical, cellular hallmarks that lead to it that's very well established are. And then I'll move on to our novel knock-in mouse model. Specifically, I'll be talking about an enzyme called tissue transglutaminase. I will often refer to it as TG2. So just a little bit more terminology with that. And then we will talk about what we can understand from the mouse model. So moving on to the main topic, which is vascular stiffness. So all biological tissue have a specific biomechanical property associated with them. The blood vessels are what is called a non-linearly viscoelastic material. So what they do is that during systole, which is when the heart is ejecting blood volume into the circulation, the aorta, which is the largest blood vessel in the body, expands like a balloon and stores the blood that's ejected and also accommodates energy that emanates as a result of this blood expulsion. And in diastole, the aortic valve closes, there's no more influx of blood, and at this point, there is an elastic recoil, which basically brings the aorta back to its original dimension. And this recoil basically, because this valve is closed, allows blood to flow only forward in a unidirectional manner. So if I were to look at a cross-sectional cartoon of this, this is what it would, the aorta would look like in diastole. It expands in systole and then of course recoils back in diastole. In this expansion, the wall of the aorta, which is the structural aspect we will be considering, is exposed to different kinds of mechanical stresses. The first is a radial stress, which happens in the radial direction. That's why it's called the radial stress. And the second is the circumferential stress that builds up on the wall, and it's often referred to as wall tension. So instead of just looking at a raw force value, we like to look at a normalized force called the stress, which is just the stress per unit area of the wall. And then we also define strain, which is the change in dimension relative to the original dimension, which you could consider to be the luminal diameter, for example. And then we can also define a strain rate, which is the time dependence with which strain undergoes change during this elastic expansion and recoil. So I talked about the vasculature being non-linearly viscoelastic. What does that mean? So basically, if you take a look at these graphs down here, you see that a linearly elastic material, basically, if you apply stress, it will generate a predictable amount of strain, and that stress to strain ratio is given by this E, which is the elastic modulus, in this case, the slope of the straight line. So that's a linearly elastic material because the stress and strain follow a linear profile. A non-linearly viscoelastic material, on the other hand, does not follow a linear profile, and biological tissues, the aorta in particular, follow this kind of parabolic, it's actually more an exponential, but it's like a curved upwards kind of profile, where you have a huge amount of strain that can be developed in response to a small amount of force being put into the system at the low phase. And then as you go higher and higher, you have to apply more force to create any deformation. You can think of this in terms of a rubber band. In the beginning, when you stretch a rubber band, it's really easy to stretch it, but the farther you go, the harder it becomes to stretch the rubber band. This is basically that type of a material. Now, in this case, we also have what is a viscoelasticity built in. What this means is that there is actually a time difference between when the force is applied and when strain is perceived. So this you can think of as a ketchup bottle or honey. When you flip it upside down, flow doesn't immediately begin, but as you whack the bottle, flow starts. This is kind of like that. It's a very viscous material, so flow does not, or the deformation doesn't initiate immediately. And for viscoelastic materials, for viscoelastic, nonlinear viscoelastic materials, the loading and the unloading curves are actually separated in this manner so that you have a loop set up like this, and this is called hysteresis. And this is very characteristic of blood vessels. So you can see what you can gain from this is that the elastic modulus, which is a constant for a linear elastic material, is no longer the case. There's no single elastic modulus, so instead we can define an incremental elastic modulus. And this incremental elastic modulus in the case of the aorta now depends on the distending pressure, which would define in which portion, how much deformation is happening, and in which part of this curve we land in vivo. Now, this nonlinearity comes in because of the known load-bearing elements of the vascular wall. The elastic part is believed to appear because of the elastin fibers that make up the extracellular matrix of the aorta. So it's one of the main components that renders elasticity to the vascular wall. And the elastin to collagen ratio also defines how elastic it is going to be at low deformation. Now, the viscous part is incompletely understood, but it is ascribed to both the cell content and the vascular smooth muscle cell tone. So I have abbreviated this as VSMC, that's the vascular smooth muscle cell. And some experiments as far back as 1964 show really beautifully the changes that happen in the vascular wall as we distend it with increasing amount of pressure. You can see that the elastic fibers are kind of undulating and squiggly when zero force is applied. But as we continue to increase the force, they become more linear and stretched out. So that's really what gives the elastic deformation curve that we see in the aorta. The other interesting thing that I would like to point out is that across a whole range of species, going from reptiles to squids, all the way to mammals like rat and even the whale, the incremental elastic modulus, when normalized to the distending pressure, pretty much lands on the same curve. So this basically tells us that the closed circulation evolved to be able to withstand and respond to a certain range of distending pressures beyond which the material will begin to fail. So, but this, I think, illustrates beautifully how evolution led to a closed circulation system that functions well for various species. So it turns out that the viscoelastic properties, or the elasticity, the incremental elastic modulus, which physiologically is often referred to as vascular compliance, is a very essential factor or property that maintains overall cardiovascular health. It's critical for overall cardiovascular health. So an aorta that is nice and compliant functions in a way so that it basically renders what's called the pulse pressure to be within an acceptable range. And how does this happen? So the main thing is that when the heart beats and ejects blood, along with it comes out a pressure pulse. This pressure pulse is going to travel forward, hit some branch points, and then come back as a reflected wave. So you can see that there will be a bunch of forward waves, a bunch of returning waves that will all kind of interfere with each other and result in this observed waveform. Now, if you have a very compliant vessel, this return wave actually reaches the heart only when systole is complete and the valve is closed. However, if the material gets too stiff, this return wave can reach the heart much earlier and during systole. And what that does is that it makes the heart work harder against this returning wave to deliver as much blood as is needed into the circulation. You can sort of think of it as trying to keep a door open against a gust of wind, to put it in some kind of like a common parlance. So now when this happens, basically we observe a huge augmentation in what's called pulse pressure, which is just the difference between the systole and the diastole. The second thing that happens when the vasculature is compliant is that it is able to take the pulsatile flow input from the heart. So your heart is beating in a pulsatile fashion. And so you'll have this huge amount of pulsatility early on. So you have high pressure during systole, lower pressure during diastole, and that's going to fluctuate in this manner in the large blood vessels that need to be compliant. But basically, through the property of vascular compliance, the aorta cushions this pulsatile effect. And by the time we reach the arterioles and the capillary, the flow gets smoothed out and the end organs do not sense this pulsatility. This is the famous term of Windkessel effect of the aorta. Now, when the vessels get stiffer, this Windkessel function actually deteriorates dramatically, and the pulsatility will penetrate much farther into the vascular tree. And as the end organs are exposed to more pulsatile flow, they're placed at a higher risk for end organ damage. So in sum, the compliance of the blood vessel minimizes the amount of work needed by the heart to maintain perfusion. It protects the end organs by rendering the final flow to them as laminar, and therefore protects them from flow-induced damage. And of course, this laminar flow also is critical for normal functioning because it allows for the optimal transport of oxygen and nutrients to the organs and the removal of waste and byproducts out back to the blood for secretion. So as you can see, if we lose vascular compliance and the vessels become stiff, we would run into a physiological issue where multiple negative effects on the cardiovascular system will be perceived. So how do we measure vascular stiffness in vivo? So the gold standard index is what's called pulse wave velocity. We have a system from Centrica where we can use a Doppler measurement of pulse wave velocity. This is a non-invasive measure, and this is a great approach because then we can really do longitudinal studies in mouse models. So we could take the same mouse and observe it from 3 months of age all the way out to say 20, 22 months of age when it's old, and really have longitudinal data to compare within in-mouse comparisons. This is just done under regular anesthesia and provides minimum discomfort to the mouse. And most importantly, there's no damage to the blood vessels. So once we are done with the final Doppler PWV measurement, we can readily sacrifice the mouse, extract the aorta and other large vessels to do any kind of analysis that we might want to do post hoc, such as Western blotting, qPCR, any molecular endpoints. So the way this is done is we use what's called asynchronous measuring. So we put a Doppler probe close to the heart, and that gives us our first waveform, and we do this along with the measurement of EKG, then you can, with a ruler, measure the distance and put the same probe at a different point and do the same measurement over again. We know the distance because we measure it, and we can then find the transit time for the pulse by comparing the two waveforms. And this distance over the time gives us the velocity of this wave in traveling from position 1 to position 2. And we can then, this pulse wave velocity then correlates to the incremental elastic modulus, or the stiffness of the vessel, through this equation called the Moens-Korteweg equation, where pulse wave velocity equals the square root of the incremental elastic modulus. And it's also proportional to wall thickness, the lumen diameter, and blood density. So this non-invasive measure of pulse wave velocity is key to understanding how blood vessels get stiffer with age. Now, I have alluded to this before, due to the viscoelastic nonlinearity of the biological tissue of the elastic modulus. Basically, the distending pressure, the blood pressure, or the mean arterial pressure really defines at which point of the viscoelastic curve we land. And so if you have a hypertensive mouse versus a normotensive mouse, then a main question that appears is that whether the increase in pulse wave velocity, which represents the elastic modulus, is due to true changes in the mechanical properties of the blood vessel, or is it merely because we're landing in a different portion of the viscoelastic, nonlinear viscoelastic materials curve, and so we're perceiving a different or increased elastic modulus. So for human subjects, a large number of studies have now developed rule of thumb corrections to account for changes in pulse wave velocity due to blood pressure. And in mouse and rat models, which would be the most used preclinical models, there's no rule of thumb correction that's been postulated yet. But what we can do is we can use an invasive index of pulse wave velocity, where we put in a pressure catheter with two transducers at a known distance apart through the femoral artery, or you could advance it from the carotid. And at the same time, we can infuse through the tail vein or the jugular drugs to increase or decrease the blood pressure. So we typically use phenylephrine, abbreviated as PE here, to increase the pressure, and sodium nitroprusside to decrease the pressure. And the presence of two transducers on this catheter allows us to calculate pulse wave velocity simultaneously while changing the blood pressure. So we get a curve that looks like this, for instance. And so this would be the invasive approach to measure pulse wave velocity. However, this is a terminal experiment, and we cannot use we cannot use this for longitudinal studies. We can only do cross-sectional studies with this. And the fact that we are invasively putting in a catheter basically means that this aorta and the femoral artery may no longer be usable for ex vivo analysis. Now, a number of studies, large-scale studies, including the Framingham Heart Study, the BLSA, the Rotterdam Study, and so forth, have shown that there is a significant increase in vascular stiffness with age. And this is actually very well summarized in this beautiful study that was published in 2010, where they considered 11,000 subjects, more than 11,000 subjects, and generated this beautiful correlation between age and hypertension. So on this axis here, the x-axis is age, the y-axis is pulse wave velocity, and the z-axis here is hypertension. And so you can see that so increasing hypertension going from optimal blood pressure to grade 2 or 3. So you can see within any age cluster, hypertension results in a higher pulse wave velocity. And within every hypertension cluster, age results in increasing pulse wave velocity. And if we consider just normal blood pressure, this data on the left shows what happens to pulse wave velocity with normal blood pressure. And you can see that with just age, even if blood pressure index is normal, there is a significant increase in pulse wave velocity. And this correlates to, as shown here, a huge increase in the risk for cardiovascular events, with the highest quartile showing a remarkable increase in risk compared to the lowest quartile of pulse wave velocity. The other thing that happens is that this stiffening coincides with the shift of the type of hypertension that individuals experience, going from mostly isolated diastolic hypertension in young individuals to mostly isolated systolic hypertension, which is the closed black bar, in the elderly. And this particular isolated systolic hypertension is actually quite difficult to treat because most of the drugs that we have in our pharmacopoeia is developed and designed to target isolated diastolic hypertension or the combination systolic diastolic. So the individuals in this particular cohort are elderly, they're prone to syncope and fall, and as they're also elderly subjects, a fall can be catastrophic with fractures, etc. And so this is a significant problem with no particular drug available for it as yet. So we note that pulse wave velocity increases with age in humans, but we do also need to confirm that the mouse model is reasonable to study this in. And here on the left are data from mice, where these are C57 black 6 mice. We aged them from birth to about 22 months of age, and we classified them into month clusters. And as you can see here, by measuring pulse wave, Doppler pulse wave velocity, which is the non-invasive index, we're able to show that in mice, there is a significant increase in pulse wave velocity as they age, with the PWV, or the stiffness index, kind of plateauing between 1 year to about 1.5 years of age, which is 12 to 15 months. But there is definitely this range from birth to old age that we can study using pulse wave velocity. The same thing is seen in rats. As we go from birth to older rats, we see the same kind of curve where there's a rapid increase in pulse wave velocity between birth to about 1 year of age, and then there's a plateau that shows up. And then for the rats, we also did the invasive index, which is a little bit easier to do in rats because of their larger vessels. And you can see here that there is, in fact, a material property change. So in the young rat shown here in red, you have a compliant blood vessel, whereas the oldest rats shown here in blue, there's a leftward and upward shift of this curve showing that these rats have significant material property change in their blood vessels. And all of the large studies have basically established pulse wave velocity as a very valuable predictor of cardiovascular risk. This index predicts future cardiovascular events even after accounting for any established risk factors such as obesity, diabetes, hypertension, and obesity, and kind of lifestyle indices and age. And the predictive value actually is, by including pulse wave velocity as a prediction index, you can improve the overall 10-year risk classification by up to 13%. And for those that are interested, this paper by Ben Shlomo and coworkers is actually a really wonderful read to look at the utility of pulse wave velocity as a predictive index. So the question then is, yes, we can agree that pulse wave velocity and vascular stiffness are an important parameter to study in humans, and develops mouse and rat models so that we can understand how to interrupt vascular stiffening. So, but before we can do any of that, we have to understand what is known about the molecular and cellular hallmarks of vascular stiffening. The main thing that's known is that with aging, there is significant endothelial dysfunction. This includes a rapid or a marked reduction in the nitric oxide bioavailability. So NO here is nitric oxide, and a drop in the endothelial barrier function. This decrease in nitric oxide bioavailability not only directly impacts blood pressure, but also regulates protein function through a post-translational modification called S-nitrosylation. It can affect protein secretion, gene expression, et cetera. And all of these molecular events have been kind of shown to happen with age. These changes are associated with VSMC, or vascular smooth muscle cell hypertrophy and hyperplasia, which is shown here in this cartoon. This is from a paper by O'Rourke and co-workers where this was a review paper where they showed in this cartoon form the changes that happen in the vascular wall. So you have the whole wall becomes thicker, you have significant amount of collagen accumulation, there's breaks in the vascular lamellar structures and so forth. The vascular smooth muscle cells undergo hypertrophy and hyperplasia. So all of this can be kind of tuned back to changes in the stiffness of the matrix due to the, with regard to stiffness, the matrix stiffness changes because there's more collagen, more elastin fragmentation, and more remodeling. Upcoming and newer studies show that the cell stiffness, vascular smooth muscle cell stiffness and tone and number of cells are also a very important predictor of the vascular wall materials, incremental elastic modulus. And all of these changes in the cell matrix and in the cells themselves propagates to what we observe as changes to vascular stiffness index, the pulse wave velocity, and subsequently vascular function. So with that introduction, I'm gonna turn it back to you, Sarah, for the audience poll, and I will take it back whenever you're ready.
Great, thanks, Lakshmi. So this poll question is, what technologies are you currently using to assess vascular aging? And it's a multiple choice, so please select all that apply. Okay, so we're just going to give you guys a few more seconds to answer, and then we'll get back to the presentation. All right, so we will give this back to Lakshmi. Thank you.
Thank you, Sarah. So moving on to the enzyme that we will be discussing today. So my lab started looking at tissue transglutaminase, or TG2, which is a known matrix remodeling enzyme in the context of vascular aging and vascular stiffness about a decade ago now. So tissue transglutaminase is a very interesting molecule. Many of you may have heard of it in the context of being a meat glue. So it's actually quite popular in the food industry to kind of change the properties of meat and to kind of make it more stiff. So if you buy really cheap steak, I'm sorry to tell you, it's made up of not steak. It's actually just meat products bound together by tissue transglutaminase. So this enzyme actually is composed of 4 domains, and it has a complex biochemistry, but at this portion of the talk, it will suffice to look at its 2 conformations. This on the left is the open conformation, and on the right is the closed conformation. Binding to GTP, shown here in red, facilitates the change to the closed conformation. So dissociation of GTP and calcium binding forces it to the open conformation. And this open conformation enzyme is what catalyzes this popular transglutaminase reaction, which basically takes the side chains on lysines and glutamines and cross-links them to form this isopeptide bond, which is basically a CONH bond on the backbone, not on the backbone of the protein, but on the side chain of the protein. So TG2 is a member of the transglutaminase family, which has 8 members. It is one of its 8 catalytically active members. The interesting thing is that actually most of tissue transglutaminase is actually in the cytosol, where it is GTP bound and rendered inactive. It is secreted through what's actually a Golgi independent and not fully known pathway to the cell membrane and outside to the extracellular matrix. And less than 20% of it is in what could be classified as an extracellular environment. And this secretion, and often its synthesis and secretion, is promoted in, for example, fibroblasts in response to activation of wound healing and fibrosis pathways. So tissue transglutaminase, actually, when we entered the concept of tissue transglutaminase in vascular stiffening, was already shown to cause the remodeling of resistance vessels. Now, compliance as a feature is more applicable clinically to the large vessels, such as the aorta and the larger kind of compliance vessels, rather than the smaller arterioles. So the same mechanisms may or may not be active in both places with regard to vascular remodeling. However, it is supportive that tissue transglutaminase mediates resistance vessel remodeling in response to both low flow and chronic vasoconstriction. The other thing that was known, which was interesting to us, was that the regulation of this catalytic function of TG2, and when I say catalytic function of TG2, I mean this cross-linking function, was significantly dependent on the availability of nitric oxide. So adding nitric oxide to tissue transglutaminase suppresses its catalytic activity. So based on all of this and the fact that not much was known about how tissue transglutaminase gets out, and knowing that in the aging blood vessel, nitric oxide bioavailability goes down, we did some studies to see if there is any change in tissue transglutaminase secretion in the vascular cell types in response to nitric oxide. And so what we did was we started with endothelial cells and either gave them a chemical nitric oxide donor or a nitric oxide synthase inhibitor. So eNOS is the primary producer of nitric oxide in the endothelial cell. And we also gave it a reducing agent, which presumably would negate any effects of nitric oxide. And what we found is that increased nitric oxide suppressed the secretion of tissue transglutaminase, and a decrease in nitric oxide promoted the secretion of tissue transglutaminase. And this happened through some mechanism that was sensitive to a reducing agent. So if we gave nitric oxide along with DTT, which is a reducing agent, the effect of nitric oxide was no longer perceived. The next thing we found was that in addition to these exogenous chemical interventions, if we merely provided endothelial cells in co-culture with either smooth muscle cells or fibroblasts, the nitric oxide from this endothelial cell layer was sufficient to regulate the secretion and activity of tissue transglutaminase in this in the basolateral cell layer, which was either the smooth muscle cells or the fibroblasts. And we show that addition of endothelial cells suppresses activity, and inhibition of nitric oxide production by the endothelial cell reversed this. We showed that the abundance or the amount of TG2 in the matrix part, in the cell-derived matrix, was sensitive to nitric oxide, and also that this likely happened through a post-translational modification called S-nitrosylation, which happens on cysteines. And so not only the exogenous provision of nitric oxide, but endothelial cell-dependent nitric oxide also regulates tissue transglutaminase. So based on this, we built a hypothesis that says that in the young healthy vessel, the eNOS-dependent nitric oxide production S-nitrosylates tissue transglutaminase and inhibits its cross-linking function. However, as we age, there is a decrease in this nitric oxide production or the bioavailability of nitric oxide, and this would lead to a decrease in tissue transglutaminase nitrosylation, leading to a disinhibition or the loss of this inhibitory effect, which would activate the tissue transglutaminase cross-linking function, resulting in increased vascular stiffness. So, so we first started off by looking at tissue transglutaminase nitrosylation and cross-linking activity, or the transamidation activity with age. And what we show here is that if we take blood vessels and they have all their smooth muscle cells, et cetera, intact, then we expose it to a decellularizing protocol so that we can retrieve the vascular matrix by itself. Then we can look at the quantity of tissue transglutaminase that's matrix bound in the native aorta. And by comparing young and old subjects, in this case, rats, on this side, we saw that the older rats have a significantly higher amount of tissue transglutaminase than do the young rats. And this was sensitive both to nitric oxide inhibition, eNOS inhibition, and the absolute removal of the endothelial layer, which would basically remove endothelial nitric oxide being available to the vascular smooth muscle. In addition, we found that in the old rats, there was a significant loss of the S-nitrosylation signal for TG2, whereas in the young rats, there was a robust S-nitrosylation signal for TG2. And so we see that this loss of nitric oxide bioavailability not only causes loss of nitrosylation, but also increased transglutaminase abundance in the vascular matrix. And we observed similar features in the human aortas from young and old subjects. And these were human aortas acquired at autopsy. So all of this is fine, but the main question then becomes, because TG2 is one of the 8 transglutaminases, how do we know that it alone is responsible for the changes in the vasculature? So we went ahead and confirmed that tissue transglutaminase is indeed the primary vascular transglutaminase. Here you see in green is the total transglutaminase activity in wild-type mice. And this is the same wild-type mouse aorta, except it's been exposed to a TG2 specific inhibitor called L682.777. And you see that the specific inhibition of TG2 practically obliterates the total transglutaminase activity, suggesting that most of the vascular transglutaminase activity arises from TG2. What we also found is that if we take wild-type mice and TG2 knockout mice and treat them with L-NAME, which is the eNOS inhibitor, we see a significant increase in pulse wave velocity in the wild-type mice, but the TG2 knockout mice are actually protected from it. And the same thing was noted when we looked at the compliance of carotid arteries using pressure myography. In addition, we did tensile testing where we basically take a piece of the vessel and pull it apart at a given force, and we can measure the force that was applied as well as the deformation that it resulted in both intact and decellularized vessels, and we can get a sense of what's going on with regard to stiffness. So if we compare the decellularized matrix, we see that the TG2 knockout mice have a more compliant vessel than do the wild-type age-matched counterparts. However, when we look at the pulse wave velocity, while there seems to be a minor trend, this is not really statistically significant. So it seems like they have a softer matrix, but somehow in vivo, we are really not perceiving that difference any longer. So the question then becomes, how does this happen? And we verified this in a couple of ways. So we wanted to make sure that the changes that we note here and here are not due to some peculiarity with their blood pressure. So when we do the invasive dual catheter technique, we see that the 2 vessels from the wild type and the TG2 knockouts are actually indistinguishable. And when we compare the stiffness of the intact vessels, we see that in vitro, so outside the body, the stiffness actually, the difference in stiffness is perceivable, but in vivo, when we look at the PWV index, it's just completely missing. So what we can conclude from this is that the vascular smooth muscle cells somehow compensate for a more compliant matrix to maintain physiological or in vivo meaningful stiffness. So then the question is, what is the mechanism that accounts for this? So while the cross-linking function of TG2 is the most important or well-characterized property of its mechanism, its activity, TG2 is actually often compared to a molecular Swiss Army knife. And this is because it has multiple functions ascribed to it. It can act as a GTPase by binding to GTP, which keeps it in the closed conformation. It can actually, through the beta sandwich region, bind to fibronectin, integrins, and syndicans and promote cell adhesion. And in so doing, it can actually change the traction forces that are developed by the smooth muscle cells and the inherent stiffness of the smooth muscle cells themselves. This is actually illustrated here, where if we look at the TG2 knockout cells, shown here in red, they are actually stiffer than their wild type counterparts. And this stiffness measure was done using this method called magnetic twisting cytometry. What we also found is that if we take the TG2 knockout vessels and we provide exogenous tissue transglutaminase, we can recover the stiffness, the passive stiffness, towards that of the wild type, independent of its cross-linking function. So provision of exogenous TG2 shifted it from this red curve closer to the black curve. And so that was actually quite interesting. Not only that, we found that the contraction response to phenylephrine was actually different in the TG2 knockout mice compared to the wild type mice. And provision of exogenous tissue transglutaminase also kind of pushed this back towards the wild type. So to understand which of these mechanisms may be playing a role, we created a new knock-in mouse model where we took the active site cysteine, which is the position responsible for its cross-linking function, and we introduced a point mutation using CRISPR-Cas9 gene editing technology, and we generated a point mutant mouse, knock-in mouse, which is expressed pretty well in all the tissues, but lacks the cross-linking function. So you can see here, compared to the wild-type mouse, the point mutant mouse has no tissue transglutaminase activity, and in fact, the levels are comparable to the TG2 knockout mouse. And we verified this using 3 different complementary activity assays just to make sure that it wasn't the peculiarity of any single activity assay. Now, it's not important to lose the activity assay. The cross-linking function, we need to maintain the other functions. And so we verified that the GTP binding is present and the fibronectin integrin binding function is also present in this mutant mouse. Now, when we now take these mice and look at vascular aging in these mice, the wild type mice go from being quite compliant to quite stiff, as is expected. We see that the TG2 knockout mice are pretty much protected from any vascular stiffening with age. And these C277S mice actually land somewhere in between. So while they do show some stiffening, it's not as dramatic as in the wild type mouse. And we can see here that this nitric oxide regulation of tissue transglutamin is actually still present. So if you take the young mice, young and old mice from wild type and these mutant mice, we see that aging actually still does increase matrix associated TG2. So the secretion of TG2 as it pertains to nitrosylation is still preserved. So the protection that's confirmed, that's produced by completely knocking out all functions of TG2 versus a single function of TG2 can now be looked at in the context of passive stiffness by tensile testing. So when we compare the young and the old vessels from the black 6 wild type mouse, we see a significant amount of stiffening. The same thing is actually noted in the C277S mice, but this looks more like a cellular contribution than does passive stiffness. And then in the TG2 knockout mice, actually puzzlingly, there's a decline in the vascular stiffness that we note with aging. And we believe that this could be because of complete loss of any matrix remodeling program in the TG2 knockout mice. Finally, the point of all of this is that looks like tissue transglutaminase plays a significant role in vascular aging, and it looks like the cellular component of contributions exist in the vascular aging phenotype. However, inhibition of tissue transglutaminase cross-linking function provides a significant protection in the context of aging. And so if we take rats and give them a non-specific transglutaminase inhibitor, and we took these rats at 18 months and gave them 3 months worth of therapy, the control animals showed a significant increase in stiffness, but the animals that were treated with cysteamine were protected from further stiffening. However, to note, these are 18-month-old rats when we started. We did not see a decrease in stiffness with cysteamine therapy, which suggests that targeting transglutaminase can prevent further stiffening, but it does not de-stiffen already stiff vessels. So with that, I will pass it over to Sarah again for the second audience poll.
Great, thank you. So this poll question is aimed at researchers in the lab, and we just want to know a little bit more about your role in the lab and exactly how you interact with your science. So if you're a clinician, a postdoc, PhD candidate, master's student, if you're the PI or a research assistant, or if you're just here to learn a little bit more about what we're talking about today. So we'll give you guys a couple more seconds to answer this, maybe about 10 more seconds, and then we'll move on. Okay, awesome, thank you. All right, so back to you, Lakshmi. Continue, thank you.
Thank you, Sarah. So, so that brings me to the conclusion of today's talk. I hope I have convinced you that tissue transglutaminase is an important and interesting target in the context of vascular stiffening with aging, and that developing TG2 therapy can be a useful way to treat aging-associated vascular stiffness. Our studies show that tissue transglutaminase is regulated by NO-dependent mechanisms, and we further showed that the trafficking and secretion of tissue transglutaminase are nitric oxide dependent, which is a significant step forward in understanding the mechanism of its secretion, which actually to date is not fully known. The studies comparing the mutant knock-in mouse with the fully knockout mouse and the wild-type counterparts show that both the matrix and the vascular smooth muscle cells are actually critical shareholders in the overall vascular stiffness that we observe in vivo. Comparing the TG2 knockout mice to the knock-in mutant mouse, we see that the mechanical interaction and the biomechanical input from the matrix is likely a key determinant of overall VSMC contributions in vivo. And then because we can actually target individual functions of TG2, it would be important to see if these can, inhibiting a single function at a time, can yield distinct changes or control over the overall stiffness, pulse wave velocity stiffness, by either just targeting the VSMC part or the matrix part or the crosstalk between the two. And most importantly, tissue transglutaminase is actually shown to be a key molecule in fibrosis, in cancer progression, in angiogenesis in solid tumors, in cardiac fibrosis, and various other contexts. And of course, TG2 is best known for its involvement in celiac disease. And so this mouse model, we believe, will be very key to study the specific contributions of the cross-linking function of TG2 to the development and progression of these diverse set of diseases that are completely unrelated to each other. So with that, I would like to acknowledge all my collaborators and students and postdocs who have put in work over the past 10 years or so to develop the talking points and the importance of tissue transglutaminase in the context of vascular aging. And of course, the funding agency that has been kind enough to support our research efforts. And with that, I'm happy to take any questions. Thank you all for your attention.
Great, thank you so much for that really awesome presentation. We are going to run a Q&A now. So at the bottom of your screen, like I mentioned before, there's the Q&A button, and if you open that up, please ask any questions that you have for Dr. Santhanam about her work and about how she collects her data. We're going to start with the first couple that came in. So the first question is, what is the potential role of TG2 in clinical aging studies?
That's the kind of hope of all of my research, is to kind of develop this to a translational end. So in the context of vascular aging, I would imagine that therapy would have to be started kind of in middle age to have significant benefit with regard to aging-related vascular stiffness. There are some very interesting studies going on in the context of cardiac fibrosis, and it would be interesting to see if interrupting TG2 function in fibrosis at different points in the development and progression of fibrosis, where interrupting it is the most beneficial. And some of those can then guide back our own clinical studies to look at vascular aging. But it is clearly a very key molecule in propagating vascular stiffness. And so we hope that this would lead to some important clinical applications.
Awesome, thank you. Hopefully that answered your question. Our second question here is, does angle correction matter while using Doppler for pulse wave velocity?
In a word, yes. So when we use any kind of Doppler measurement, the frequency shift is proportional to cos theta, with theta being the angle at which the probe is relative to flow, blood flow. And so you do want to keep that as close to zero as possible to prevent any Doppler shift. The closer you get to 90 degrees, the worse and worse it's going to get. And so the ideal thing would be to place the probe in the direction of the flow.
Great. So we do have a couple more questions here. This next question is, where are the TG2 expressed? Are they expressed in all tissues?
So TG2 is very pleiotropic and highly expressed enzyme. It's expressed in a number of places. It's been shown in the kidney. It's been shown in the heart, liver, lung, blood vessels, and so forth. So it is highly expressed in a number of organ systems. However, the important thing to keep in mind is that it's not its expression, but its dissociation from GTP and secretion to the outside the cell, to the matrix environment, which is what regulates its activity. So what happens is that TG2's activity is actually held latent during just homeostasis. And if you have something like like an injury accompanied by wound healing or a profibrotic insult, that's when TG2 gets activated and secreted. And so its activity is perceived in a bunch of other tissues.
Great. We have another question here. So this question is, are there human genetic variants that affect TG2 function?
Yes, there are human genetic variations that affect TG2 function, and there's a number of SNP studies that were published by a group in Europe that are very fascinating. And I'm not an expert in that, in the SNP variations of TG2, so I would point you to those studies. But yes, there are genetic variants, and it's very intriguing to see how or which mutations may lead to either suppression or augmentation of its activity in vivo.
Great. We have another question here that is asking about cellular senescence. So this question is, any insights on the roles of cellular senescence in vascular aging?
That is also a very intriguing question. So it is believed that the VSMCs senesce, and often we see a decrease in the VSMC content in the very, very elderly. So there is some kind of a senescence that happens in VSMCs. So the question with regard to does TG2 affect that, that's actually interesting. We have not done that, but those are definitely studies that we could do, particularly as we have in hand extremely aged animals from both TG2 knockout and the mutant knock-in mouse. And so this is something we should definitely look at. So thank you for that suggestion.
Mm-hmm. Great, and we have time for one more question. And so this question is, how does TG2 cross the plasma membrane when being secreted?
So the mechanism of secretion is still not fully understood, but it is believed that there are, that it's an endosomal transport perhaps, or exosomes. So the exosome endosomes basically cycle it to the cell membrane and back in, et cetera. So that's the most prevailing mechanism for TG2 secretion. It does not happen through Golgi, that much is known. So our hypothesis is that nitric oxide may control this, especially in the vascular tissue, secondarily by regulating what is called NSF, which is the N-ethylmelamide sensitive factor. And if you have nitric oxide in abundance, the NSF-dependent secretion actually is suppressed. And this was shown by Charlie Lowenstein and co-workers. It's a really beautiful Cell paper back in the early 2000s sometime. So this NSF mechanism may be active to support exosome formation, et cetera, to help TG2 run through the cytosol and out into the extracellular environment.
Great. Well, thank you for that. Thank you for all of your questions that you just submitted. We have reached the 1 hour mark for our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any outstanding questions privately with anyone waiting for a response, and then we will also publish a written transcript of all questions and answers from our Q&A session today. I'd like to thank Dr. Santhanam again for her interesting and incredibly informative presentation. I'm hopeful that you all walk away satisfied today, that we met your learning objectives, which were to understand the changes in pulse wave velocity with age in mouse models, to determine the specific role of TG2 in vascular aging, and to evaluate the role of the vascular matrix versus the vascular smooth muscle cells to overall in vivo stiffness described by pulse wave velocity. Please feel free to reach out to us directly should you have any more questions about the methodology used to acquire the in vivo data presented today by Dr. Santhanam. Please also do not hesitate to direct any questions to us that are for her because we can forward them along to her as needed. Thank you again for all of your time today and taking the time out of your very busy schedules to attend our session. We look forward to seeing you at a future Syntica instrumentation event, and I hope everyone has a wonderful day. Thank you.